Good morning to you all. If you remember, we were here last time. Do you remember this? MSA, PEA, and CAN. And we said that these are the selective media. And the one that is most commonly used is MSA, mannitol salt agar. PEA and CAN, not very much used. But MSA, almost in many labs. We talked about these last time. Also, we talked about this. And this is again, we can repeat this, that is mannitol salt agar, where the salt, 10% salt is an inhibitor, makes it selective. But the Staph aureus is halophilic. What is halophilic? It can tolerate salt. Plus, we have phenol red as a pH indicator, and this makes the media change from red, original color, red like this, into yellow through this pH indicator, because the S aureus, like any bacteria, it produces waste, and this waste is acidic and changes the pH. So change the color from red to yellow. So if like this one here, this is totally yellow, although the original is red. So this means that this is what? Staph aureus. Here you see growth, but no change in color. This means this is a staph, but not aureus. It can be epidermidis, it can be saprophyticus. No growth. Here there is a growth with change in color. There is a growth with no change in color. Here there is no growth and thus no change in color from the first place. So there are some researchers who work only on a certain bacteria. Some people work only on staph. So they are specialized in staph. In order to save time and effort, they select a media that from this media you can tell this is Staph aureus or not. Like media called chrome agar. What is a chrome? Chrome means color. So chrome agar, Rambach medium. This medium is, you can see the color here, it's mauve colored and contains cefoxitin. Cefoxitin is an antibiotic. And this antibiotic also, it can make distinction and differentiation between MRSA and non-MRSA. You know, last time when we talked about MRSA, methicillin-resistant Staph aureus. It can resist many antibiotics and difficult to treat. This is the type of S aureus which we don't like and we don't want to have in our hospitals and houses. So this media contains the color plus contains the differentiation antibiotic, which is cefoxitin, and it gives also colors. So the mauve color colon is here. Chrome agar is available for many organisms. There is chrome agar for E. coli. There is chrome agar, for instance, gram-positive bacilli. The idea about chrome agar is that it makes life easier for you, that if you are looking for Staph aureus, it will give it in a certain color, like mauve color. This is a mauve color. Companies come up with ideas for two reasons: to make life easier for you as a microbiologist and to make profit for the companies, both things. Incubation. Incubation, we need temperature, and we want also the oxygen and CO2 requirements. In particular for Staph aureus, that's what we need. Look, we need 35 degrees centigrade, or 37 if you like, 35 plus minus, so 37. You need oxygen, facultative anaerobe, and also sometimes it's much better if you want to display hemolysis in a good way, you have to use CO2. It appears better. Even in anaerobic conditions, no oxygen, hemolysis is displayed even better and better. And you need only 24 hours. Some selective media like MSA, they require more than 24 hours to give the color, because the color development is slow. Now let's see the colony morphology. How do colonies appear on the plate? Of course, here we have a standard plate, which is 5% SBA, sheep blood agar. On this plate, give description to the colonies, the morphology of the colonies. Are they big? Are they small? Are they yellow? Are they white? Are they creamy? Just description. And these descriptions don't expect that you are going to give accurate diagnosis from the colony morphology, but it can give you a hint. It can just give you a clue of what's going on. So look here. If you remember in the basic micro, we gave you this shape, every bacteria. You have to look at the shape. You have to look at the elevation, high or low. Margin, is the margin like a circle or is it wrinkled? The size, big or small. Color, sometimes they give you different colors, white, yellow, creamy, green, red, sometimes like Serratia, and optical density. Is it colorless? Is it very dark? Opaque, we call it. Let's have a look at the Staph aureus. What we are going to talk about here is in most cases. So shape is like this one, so circular. All the time is circular. It's not irregular, it's not filamentous, it's not rhizoid. It's just a circle, complete circle. Of course, inside this circle does not represent one bacteria, does it? It's a colony with millions of bacteria inside it. Okay, so we call it colony, CFU, colony forming unit. It's circular. Elevation, raised to convex, little bit high. It's not flat, it's not crateriform, it's not ambulate. It's raised or little bit convex. So slightly raised or low convex. Margin, it's entire like this, entire. So the margins are entire, not undulate, not filamentous, like the shape and the form here. Not lobate, loops, not serrate like teeth, filamentous. So that's the margin, entire and smooth, totally. Size, medium size. You are not going to just hold a caliber or just hold a ruler and take out the plate and start measuring. No, this is not the idea. The idea is that with your normal eye, you can tell that this is pinpointed, very small. You can tell that this is big. You can tell this intermediate. So with naked eye, no ruler, no mobiles, nothing. Just with naked eye, you can tell, this is pinpointed, very small. It looks like strip. So medium to large, it's not very small or pinpointed like strip. It's yes, moderate to large. Color, creamy to yellow. Sometimes creamy alone, the Staph aureus. And sometimes creamy to yellow. I will show you in a minute. And beta hemolytic. Staph aureus gives beta hemolytic. If you want to see the good display of beta hemolysis, just incubate it at CO2 or maybe anaerobic. Optical, opaque. Opaque means you can't see anything through. We have transparent, just like glass, you can see through, that's transparent. And you have opaque, and we have something in between, which we call translucent. You can see just a little bit. Here it's opaque, very dark. Very dark doesn't mean it's black, but you cannot see through it. So these are 1, 2, 3, 4, 5, 6 characteristics that give us the shape and the morphology of the colony. These here come through experience. You have to see S. aureus many times in a lab with good load of work. That's S. aureus. Let's see how it looks. See, this one looks like creamy. Is it white? No. Is it yellow? No. Sometimes it's creamy like this or creamy yellow. And look at this. What's this? Hemolysis. And the hemolysis is really clearly displayed. No blood here. It means that the bacteria have eaten all the blood, leaving no blood here. We call this hemolysis. For those, yes, who were not in the lecture, we had alpha hemolysis. The color is green, which is partial hemolysis. We have full hemolysis, complete hemolysis. It's beta hemolysis. And we have no hemolysis or very slight, which is gamma hemolysis. Creamy yellow, convex, circular, you see, entire, it's just complete, and sometimes glossy, it shines a little bit. This is how it looks, Staph aureus on MSA. See, still there is traces of red from the original color of the plate, but here it's yellow. Here is yellow. Here's no growth, and there is growth without yellow, still red. This is aureus. This is epidermidis. This is unaniculated. Let's go to the epidermidis. Staph epidermidis, which is on the skin of everyone. That's how it looks, circular. All the staph looks circular. All the staph colonies are circular, and they are all entire. Raised and convex, just like the staph aureus. Entire, also just like the staph aureus. And here, small to medium, not moderate to large. This is smaller than the S aureus. And as I said, you need experience in the lab to be able to judge it, to judge the size of it. Experience needed here. Gray-white, like creamy. Non-hemolytic, so it doesn't hemolyze blood like the S aureus. And it's opaque. Sometimes the staph epidermidis, S epidermidis, it's slimy and sticky and adheres to the media. So if you take lube and just try to move it, it's going to be sticky and maybe it doesn't even move with you. Let's have a look. That's how it looks. It doesn't look like the S aureus, if you remember. Yeah, in the aureus just seconds ago, doesn't look like the aureus. Doesn't. It's even smaller, and the color is a little bit different. Gray-white. There it was creamy. And sometimes the aureus is creamy yellow. Sometimes. Okay, smaller than the staph aureus, and convex and circular. And the last one of the three that we are interested in is the staph saprophyticus. This is not like the epidermidis. Epidermidis is totally normal flora, microbiota. So it's on the skin of every person. This one, no. This might affect the sexually active ladies and cause UTI. Shape the same, circular. Elevation is convex, little bit higher than the other two, epidermidis and staph aureus. Margin entire like this. Size, it's large, so it's larger than the epidermidis and sometimes even larger than the aureus. Color creamy yellow. Beta hemolytic, like the staph aureus. Opaque and very glossy. What is glossy? Shiny, exactly. Glossy is shiny. So very glossy. And butyrate. Butyrate is like butter-like. You know what the butter is? Yes. So when you just take it, let's say you have it on the plate, SPA, 5% SPA, and with the lube, it cuts like butter. So it's butyrus or butyrate. That's how it looks. See? The colonies are large and have some yellowish color. Now let's come to the diagnosis. Gram stain. Gram stain is a cornerstone, and it's #1 test that comes on top of the list is gram stain. Because if you don't do gram stain, you will get lost right from the beginning. With gram stain, it can give you an indication where to go. Let's say gram positive or gram negative. Gram negative have a different battery of tests than gram positive. Cocci and bacilli, they have different tests. So very important is to start all the time with gram stain. So gram stain. And it's presumptive. It doesn't give you the exact diagnosis. From gram stain, if you see gram positive cocci, you cannot tell this is staph aureus, this is staph epidermidis, this is micrococcus. Maybe micrococcus is bigger, but it's just presumptive. Presumptives mean give you a good idea, not exact idea. Morphology of colony. Again, it's a presumptive. As we said, colonies, large, small, pinpointed, yellow, creamy, glossy, opaque. These things, they give you a hint on the way of complete diagnosis. Then catalase. Now we come to the real business. Catalase. See, bubbling. What is the cause of this bubbling? 2O2. H2O2 into water, H2O, plus H or O2, O2. This is O2. So the bubbling here is a result of the oxygen and the breakdown of the hydrogen peroxide, H2O2. And this is also, it's a key test. By gram stain, comes after the gram stain directly. So if it's catalase positive, we can rule out that this is strep. We can say now we go for the staph. If it's positive, if this is catalase positive, what would I do next? I go for the coagulase. If coagulase positive, then this is what? Staph aureus. So gram stain, catalase, and then coagulase. Coagulase positive is Staph aureus. But what if it's coagulase negative? Strip, no, it's not strip. Strip, we could tell strip from the catalase. But here, if it's not, then we have to go to two tests. Coagulase negative staph, epidermidis, and saprophyticus, or it's micrococcus. How could we tell? We have two tests. Microdase or modified oxidase. Micrococcus is modified oxidase positive. The companies, they call it microdase. And this is how it looks. It's purple on a white paper. We do it on a filter paper. If it's positive, then it's micrococcus. If it's negative, epidermidis or saprophyticus. What do we do? The best test, which we talked about last night over the Zoom meeting, novobiocin. We make the novobiocin, and then if it's sensitive, epidermidis. Resistant, saprophyticus. I will show you all what I've talked about in the form of a flowchart in a minute. Don't get puzzled. Furazolidone, that's another test, not very much done. And there is also bacitracin test, 0.04 units. Bacitracin test is much better or mostly done for strip, but you can use it here in staph, and I will show you when. We can add just novobiocin. This is just a table that illustrates and summarizes what I have talked about. Catalase. So all staph are positive. Micrococcus is also catalase positive. Yes. Strep, Enterococcus, forget about the others. Microdase, which is modified oxidase. Staph, negative. But micrococcus, positive. This is the only one positive. Forget about the micrococcus and just focus on this. So modified oxidase, which is microdase, 6% reagent, then this is positive. Aerotolerance, FA means facultative anaerobe, staph, with oxygen or without oxygen, FA. Micrococcus, only oxygen. Strep, facultative. Bacitracin, strep is the most one. That's why bacitracin is mainly for strep. The same here. Clumping factor and coagulase test. Coagulase test. Which bacteria gives coagulase positive? Staph aureus. There are others, but we are not interested in them. Staph aureus here. If you see Staph aureus, and that's all. We don't want you to have Staph aureus anaerobius. No, just Staph aureus. Log the intermedius. See? Variable. Positive. Hiatus variable. This is the one we are interested in. And about these here, maybe you just have to look at them just like this. There's a nuclease test, there's alkaline phosphatase, there's ornithine decarboxylase, novobiocin, this is also important, novobiocin, polymyxin B resistant, pyre, that's for strep, but we can use it also for staph, mannitol, maltol, sucrose, mannitol, hemolysis, that's new, hemolysis. Which one makes hemolysis directly? Staph aureus, epidermidis now. That's another one, saprophyticus. Urease, alkaline phosphatase, trehalose. These tests, novobiocin resistant, but these are not used except for reference labs or research labs. Epidermidis, look, the same. But no one will use, in a normal lab, routine lab, no one will use urease for epidermidis, although it's strong positive. This is a flow chart. Based on the tables which I showed you, based on these tables, we have made two flow charts. One flow chart that contains all the tests, and one flow chart that is practical for normal routine diagnostic lab. Let's see both flow charts. So first, we make gram stain. It's good to make wet mount. What do I mean by wet mount? Just slide, normal saline, or distilled water if you like, and under the microscope. Why do you think we do this? Sometimes you look at the colonies and you think they are bacteria. in the first 24 hours. But they are in fact yeast, Candida albicans, and they look like staph. Many of us made this mistake, and we didn't do gram stain, and we just started catalase, coagulase, this and that, and just wet mount, it showed us that it's Candida albicans. And usually Candida albicans under the microscope, it's bigger, larger, quite larger than the bacteria, because the bacteria is very small, but the Candida albicans, the yeast, is very big, and it has a greenish fluorescent color, not like the bacteria. So you can tell exactly when you look under the microscope, this is yeast. It's not staph. So you stop and you report yeast, and you don't go to the other tests. But if you make gram stain, you can tell. But many labs, they don't do gram stain, and they fall in this mistake, and they spend a day or two working and working, and then all the work is false. A wet mount could have solved the problem right from the first place. So gram stain, gram positive cocci, gram positive bacilli, and gram negative. So gram positive bacilli, it's something else. We'll talk about this in later chapters in this course. The gram negative, it's the next chapter, or the chapter after the next one. And if it's gram positive cocci, then you go down like here, as shown here, catalase. So gram stain and catalase come in the first, or on top of the flow chart. Catalase negative, then this is what? Strip. Catalase positive, staph. Staph or micrococci, to be honest. Here we make coagulase, and some people just make MSA, the culture plate, MSA. Red to yellow, then Staph aureus, and finished. Some people who don't have budget in their labs, they just do coagulase. And if coagulase is positive or MSA is positive, then it's S. aureus, which is the one that's most pathogenic and the one we are interested in. There are others, intermediates, Hyacus, Liferide, Delphini, not very much interested. But you have to know that S. aureus is coagulase positive, but it's not the only one. Then if it's negative, coagulase negative, and MSA negative, then we do pyre or novobiocin. Pyre, we will talk about it in the second lab in chapter 2, because pyre is a test mainly used for strep. Yes, catalase negative. Novobiocin, we can use it here. I mean, you have 4 options here: glucose, alkaline phosphatase. As I told you, this is for reference lab or a research lab. We'll use lots of tests. In a normal lab, normal diagnostic lab, we don't use all this panel of tests. Maybe novobiocin, and that's all. If it's sensitive epidermidis resistant, then it's going to be saprophyticus. But pyre to be included here, it gives you very detailed diagnosis. Look, if it's pyre negative and resistant, it's this. Pyre negative and sensitive, you have to do alkaline phosphatase. And imagine if you do this, it's going to take 3-4 days. If it's pyre positive and resistant for glucose fermentation, Escone and xylosis, and so on. So here, pyre and novobiocin. Normal labs, novobiocin and enough. I will show you in the next flow chart. In a research lab, diagnostic lab, reference lab, you can use both, because then if you use both, you have 4 options. And each option, you need another test also. And this is not all. Look at the next slide. Yeah, that's continuation of the first one, alkaline phosphatase. Then positive, negative. Then you have this, POLB. So this is for a reference lab, ornithine, alkaline phosphatase. See, this is just for a reference lab and a diagnostic lab and a research lab. This one, not very much recommended for normal, routine, quick diagnosis. The one we recommend in normal labs is this. Look, for simple routine work. Gram stain, look how simple it is. And this is the one I need you to have in your brain. Gram stain, wet mount, just to rule out yeast. Gram positive cocci, gram negative, gram positive bacilli. Leave it because each one of them has its own flow chart. Gram positive cocci, catalase, Then if it's negative, it's going to be strep, positive, staph and micrococci. See, staph and micrococci. Strep, they have their own battery of tests, profile of tests. Leave them to the next chapter. Staph and micrococci. What do we do? Coagulase or MSA. Coagulase is more readily available to the labs. See, if it's coagulase positive and MSA positive, then Staph aureus and finished. I don't want to give you the others. Here, if it's negative, coagulase negative, oxidase, modified oxidase, which is microdase, and bacitracin. Usually people use this. So if it's oxidase, modified oxidase positive, or bacitracin sensitive because it's a disc, like novobiocin, then it's micrococcus. Modified oxidase is enough. Then if it's modified oxidase is negative or bacitracin resistant, then you have to do the novobiocin. Sensitive is going to be the one on the skin, epidermidis. Resistant, saprophyticus. This is very quick and can be good, and it will do enough for a routine diagnostic lab. Not like the one I showed you earlier, which contains many tests and which will take time. And it's only, as I said, for research and inference lab. And there they have kits, or they have commercial kits that do everything in 1 hour, all these tests. You don't have to do the test separately, one by one. Okay, serodiagnosis. Serodiagnosis is of no value to the staff for 2 reasons. One, it's because of cross-reactivity, which means that there are microorganisms or other bacteria that have similar antigenic determinants. So when you want to test them, these other bacteria, they give you positive results exactly like this S. aureus. So we have cross-reactivity. Another thing is that the Staphylococcus infections caused by Staph aureus, they don't produce protective antibodies, which you can detect easily in serology. So for these reasons, it is no value. 7, other identification methods which are very effective, but expensive and not very much used, is this MALDI-TOF, MS, mass spectrometry. It's matrix-assisted laser desorption ionization time of flight mass spectrometry. This is a method, a machine, that where you take from the culture, and within a period of time, you will get AST, antimicrobial susceptibility testing, identification, and even subtyping. And maybe sometimes they can give you the genes which make S. aureus resistant. Okay, so here we have AST, antimicrobial susceptibility testing, or antibiotic, or sensitivity testing. AST. We have here what we call the MIC-A gene. What is MIC-A gene? MIC-A gene is in certain Staph aureus, which causes what? It causes the bacteria to become resistant and to produce PBP. What is PBP? It's penicillin binding protein, which is an enzyme. What does this do? When you take the antibiotics as a treatment, if this protein, PBP, is there, it will break down the penicillin. And then the penicillin, which is an antibiotic, will become no longer effective. So it breaks down the antibiotics, and then your treatment will fail. That's why we call this is the product of the MIC-A gene, which produces this protein. And look what this protein makes. This is penicillin, and you have the red square in the middle here, which we call beta-lactam ring. And the PBP and breaks this red square. And when this is broken, the penicillin becomes 2 pieces, and it becomes ineffective, not effective. And then the treatment will fail. That's what the MIC-A genes. So it produces PBP, which breaks beta-lactam ring, and the antibiotic becomes ineffective. And the bacteria that contains this MIC-A gene, what do we call it? MRSA or MRSA, methicillin-resistant Staph aureus. MRSA has 3 types. One, CA, which is the one in the community acquired, the one among us. And then we have the LA, livestock. What is livestock? Cattle, sheep, poultry, chicken, and so on. Things that animals that you raise at home for a benefit. That's livestock. Among animals. Can it jump to the human beings? There is a possibility. It can jump from the animal to the human being, especially if you're a farmer. HA-MRSA, hospital acquired, which we call, in other words, nosocomial infections. So we have 3 types. There is Visa and Versa. What is Visa? Another antibiotic, not methicillin. It's vancomycin intermediate susceptible S. aureus. Not very common like MRSA. Visa and Versa, vancomycin resistant S. aureus. Here, for AST, we have an international institution that can take care of the AST. We call this Clinical and Laboratory Standards Institute, CELSI. CELSI is an American institute that gives us every year or every 1 or 2 years an update of the antibiotics. Are the antibiotics effective against S. aureus or not? How? We will talk about this in details in the lab. Of course, there are 2 institutions, one American, CELSI, this one, and another one is European. We call it UCAST. We'll talk about these 2 and AST in details in the lab. Prevention. How do we prevent a Staph aureus infection, particularly MRSA? First, we have to know that there are no vaccines against SA. Treat carriers, health team. As we said at the beginning of the chapter, that we have medical team who have the S. aureus as microbiota or normal flora in their nares. And there, these people, it's normal to them, but they can infect you. Therefore, we have to treat them. Third thing, so bathe the newborn with 3% hexachlorophane. That's a disinfectant or antiseptic. In the hospital, when the mother delivers the baby, as long as the mother is in the hospital, the nurse is there, they have to bathe this newborn with this disinfectant or with this solution, 3% hexachlorophane. Why? So that the baby's or the newborn's skin is free of S. aureus, particularly MRSA. Otherwise, this baby will be infected and will infect people at home. Fourth thing, treat all MDR bacteria. There are, what is MDR? Multi-drug resistant. We have MRSA, we have Pseudomonas aeruginosa, we have Acinetobacter baumannii, and others. All these are MDRs. Bacteria difficult to treat. We have to treat all these people who have these bacteria. And finally, screen admitted patients for MRSA. Many hospitals, they do this. When you are a new patient and you are a new admission in the hospital, first thing they come to you is they take maybe nasal swabs, rectal swabs, many swabs from your skin, and they make culture just before you enter. And then this culture, if it shows that you have MRSA, then they isolate you and they treat you differently and in a special way. We call this screening. So these 5 points, they are related to prevention of S. aureus in general and MRSA in particular. The end. Finished with this chapter. Stop. Okay, we stop here. And if you have any questions, I can answer. If not, see you next lecture. Any questions, ladies and gentlemen? No questions? See you next time.